Hello viewers, you are welcome to my YouTube channel Botany. In this episode, I would like to discuss on APG system, a modern system of classification of angiosperms. APG system of classification that uses a DNA sequencing and other molecular data. A landmark paper published in 1993 by Mark Chase and 41 co-authors with an analysis of 500 flowering plants is the base for the APG system. The first version of the system was published in 1998. Three further versions of the system of classification were published in 2003, 2009 and 2016. Unlike older systems that relied on morphological characters, it is based on phylogenetic relationships which reflect the evolutionary history of the plants. It focuses on the monophyletic groups, which are groups of the organisms that share common ancestors and all its descendants. The system abandons the traditional ranks like class and order in favor of informal groups like clades. It provides a more accurate and natural classification of plants than the older systems. The system has led to the better understanding of the plant evolution and diversity. The APG system represents a significant advancement in botanical classification. While it has certain limitations, its merits far outweigh the demerits, making it the preferred system of classifying flowering plants. The adjoining diagram shows an overview of the extant Angiosperm phylogeny and classification based on APG4. The contents of this video: APG system, a historical background, the development of APG system, core principles of the system, limitations of the earlier systems, clades and cladograms used in APG system, the case studies, merits and demerits of the system, and lastly, references. In early 90s, the first large analysis of the flowering plants based on DNA sequences were published. These had become possible due to major developments in DNA sequencing technology and computing power in the late 20th century. Flowering plants were the first major group on which a large group of scientists collaborated in a comprehensive analysis of this type, collecting sequences for the same genes so that the data could be combined. In 1993, a landmark paper with the analysis of 500 flowering plants was published by Mark Chase and 41 co-authors. This paper was based on the sequences of one of the major genes involved in photosynthesis, and the analysis involved the botanists working with the computer programmers because the program had to be rewritten to allow them to analyze such a large set of data. A major difference between the resulting paper and all previous classifications was that it was a broadly collaborative effort with 3 compilers and 26 contributors from 5 countries. Rather than name it after the compilers, the decision was taken to use the name of the larger group, and the new system was named the Angiosperm Phylogeny Group of Classification, APG for short. The first version of the APG system was published in 1998. 3 further versions of the system of classification were published in 2003, 2009 and 2016, each with multiple compilers and contributors. APG1 was the first version, which was published on a comprehensive analysis of molecular data primarily from the chloroplast DNA. APG1 classification of angiosperms recognized 25 orders and 79 families. The second version of the APG system incorporated additional molecular data and revised the classification of several groups. It recognized 41 orders and 175 families. The third version of the APG system included further revisions based on new molecular data and recognized 44 orders and 163 families. The fourth version of the system has 10 compilers and 15 contributors from 6 countries. It incorporated even more molecular data and revised the classification of several groups. APG4 recognized 64 orders and 247 families. Each version of the APG has included a list of families or the genera of uncertain position. In 1998, 25 families fell in this category, but by 2016 the list has shrunk to 7, demonstrating the increase in knowledge as more plants have been sampled and more DNA regions have been investigated. The APG system, which emerged in the late 20th century, represents a significant advancement in plant classification. It is based on phylogenetic relationship inferred from the molecular data, primarily from the DNA sequencing. The core principles of the APG system includes the APG system is rooted in the concept of phylogeny, which is the study of evolutionary relationship between the organisms. By analyzing the genetic data, the APG system aims to reconstruct the evolutionary history of the angiosperms. It relies heavily on the molecular data such as DNA sequencing to infer the phylogenetic relationships. This provides a more objective and accurate basis for the classification. The system emphasizes monophyletic groups which include common ancestors and all of its descendants. This ensures that the classification is based on a natural evolutionary relationship. Cladistics is a method of classification based on shared derived characters. There are traits that are unique to a particular group of organisms and their descendants. By identifying shared derived characters, the APG system can group plants into clades that reflect their evolutionary relationships. The system uses informal groups such as clades instead of formal ranks, that is class or orders. This allows for a more flexible and adaptable classification system that can accommodate new discoveries and changing understanding of the plant relationship. The system is continuously evolving as the new molecular data becomes available. This ensures that the classification remains up to date and reflects the latest scientific understanding. Older classification systems faced several limitations that ultimately necessitated a more accurate and reflective approach. Many older systems were based on artificial characteristics. These characteristics often did not reflect evolutionary relationships, leading to inaccurate groupings. Early systems did not explicitly consider evolutionary relationships, which are fundamental to the understanding of the diversity of life. This limited their ability to provide a coherent framework for classifying organisms. The classification of the organisms often relied on subjective judgments about the similarities and the differences. This could lead to inconsistencies and disagreements among the scientists. Older systems were based on limited data, primarily morphological observations. This made it difficult to accurately determine the relationship between the organisms, especially those with similar appearances but distant evolutionary histories. As our understanding of the evolutionary relationships has improved, it has become clear that many traditional groups, such as Monocots and Dicots, were not monophyletic. This means that they did not include all of the descendants of a common ancestor. So this is the cladogram showing the position of monocots and eudicots within the angiosperm as per APG4. Most of the species we would previously have called dicots now fall in eudicot category, though the separation of the 5 groups within the core angiosperms in the diagram. The monocots, in contrast, are monophyletic lineage, comprising all the descendants of the common ancestor. The magnolids and its relatives and so-called basal angiosperms are diverged before the monocots, previously thought to be the oldest lineage. The angiosperm phylogeny group or the APG system utilizes various clades to classify the flowering plants based on the evolutionary relationships. Here are some of the clades recognized in the APG system. Monocots, which includes camelinids, liliales, asparagales, alismatales. Eudicots with ranunculales, Eudicots, rosids, asterids, caryophyllales. These are just a few examples of the many clades recognized in the APG system. The system is constantly evolving as new data becomes available and the classification of the plants may change accordingly. These are some of the case studies which illustrate the significant impact of APG system on our understanding of plant diversity and evolution by providing more accurate and reflective classification of the plants. Some of the case studies include reclassification of monocots and dicots, recognition of new plant families, merging of existing families, and change in plant relationships. With reference to the reclassification of monocots and dicots, which is one of the most significant change. So previously, the monocots and dicots were considered to be two major groups of the flowering plants based on the number of cotyledons. System recognized that the monocots are monophyletic group but dicots are not. Instead, the system divided the dicots into several monophyletic groups including the Eudicots, which comprise most flowering plants. This reclassification had a profound impact on our understanding of the plant evolution and diversity. The APG system has led to the recognition of several new plant families. For example, the family Boxaceae, which includes boxwood plants, which was previously considered to be a part of Euphorbiaceae. However, the APG system recognized Boxaceae as a distinct family based on the molecular data. This classification has helped to clarify the evolutionary relationship between the different groups of plants. The APG system has led to the merging of existing plant families. For example, the families Crassulaceae and Saxifragaceae were previously considered to be separate families. However, the APG system recognized that these two families were closely related and merged them into single family, that is Saxifragaceae. This merger has simplified the classification of the plants in this group. The APG system has also led to the significant changes in our understanding of the plant relationships. For example, APG system has revealed that water lilies are more closely related to the magnolids than other flowering plants. This finding has challenged the traditional views of the plant evolution and has provided a new insight into the early diversification of the Angiosperms. The Angiosperm phylogeny group system has revolutionized the classification of the flowering plants, offering numerous benefits but also presenting certain challenges. These are some of the merits and demerits of the system. The merits includes the APG system is firmly grounded in phylogenetic principles, reflecting evolutionary relationship between the plants. This provides a more accurate and natural classification. The system relies heavily on molecular data such as DNA sequencing, which offers a robust and objective basis for the classification. The system focuses on monophyletic groups, ensuring that all the members of a group share a common ancestor and all of its descendants. The use of informal groups such as clades allows for a more flexible and adaptable classification system, accommodating new discoveries and changes in our understanding of the plant relationships. The system provides a standardized framework for classifying flowering plants, facilitating communication and research among the botanists worldwide. The system has a practical application in the fields such as horticulture, conservation biology, and agriculture. Here are some of the demerits of the APG system. The APG system can be complex, especially for the non-specialists due to the large number of clades and the use of technical terminology. The new molecular data becomes available, the APG system may require revision, leading to the changes in the classification. The reliance on the molecular data can be limited by the availability of the fossil evidence. which can be challenging to interpret for some groups of the plants. While the APG system is largely objective, there may be instances where subjective judgments are required in determining the relationship between certain groups of the plants. The APG system represents a significant advancement in botanical classification while it has certain limitations. Its merits far outweigh its demerits, making it the preferred system of classification of flowering plants. The viewers may visit these websites for detailed information on APG 1, 2, 3, and 4 versions of the classification systems. Thank you.